Kia ora koutou, ko Erika taku ingoa, ke manaki whenua ahau I mahi ana. Hi, I'm Erika Hendrikse, and today I'm going to talk about the path forward to precision pest control through novel toxin development. The work I'll be talking about today is part of a project that's been led by Brian Hopkins at Manaki Whenua. I've been contributing as a postdoc. This work is very much in progress, so I'll be presenting mainly our research approach, but also some preliminary data. So first of all, why do we need to develop species-specific toxins? So as someone who is not an ecologist, I did know that introduced mammals were a major issue for New Zealand wildlife, but I didn't realize just how critical this is. So I've now learned that it's not just rats, possums, and stoats, but also hedgehogs, feral cats, mice, wallabies, and others. And as part of this project, I've learned a lot about toxins and how they can be really effective in pest control operations, especially compared to traps, which are not as good for landscape scale pest control. But what really surprised me is the limited options of toxins that we have at our disposal. And ultimately, an expanded toxin toolbox could help us to achieve our predator control goals in New Zealand, like Predator Free 2050, particularly as pest control moves from remote offshore islands to more populated areas. So if we had a magic wand and we could wish for a new toxin, then we'd probably want it to have these properties. It would be highly effective and it would be specific for pests. It would have improved animal welfare properties and negligible environmental impact, as well as suitable chemical properties for manufacture. So in this project, I'm focusing on the aspect of specificity. So by that, I mean species specific or specific for a family of related animals, for example, mustelids or marsupials. And I think this diagram helps illustrate why species specificity is important. This is the Swiss cheese model of risk management. You may have seen it around for COVID risk diagrams, and I've repurposed it for toxin use in pest control. So our current toxins are relatively broad spectrum, so we put these barriers in place to protect our non-target species, so wildlife, but also livestock, pets, and ourselves. For example, we can reduce access to bait and bait stations, or make the bait less attractive to non-target animals. So while we can optimize these barriers further, no barrier is 100% effective all the time for all species, and they can add costs or complications. So if we were able to design toxins that had built-in specificity, these would become extra barriers that we could add that were intrinsic to the toxin. And the reason I've added 2 layers of cheese here is because toxins can be species-specific in 2 broad ways. So they can differ in their toxicokinetics, which is related to how drugs are absorbed and processed in different animals, or they can differ in the efficacy of their molecular mechanism in different animals. And this is what I'm focusing on. So that's why we need new toxins, but the challenge is how we're actually going to develop a new highly specific toxin, given that there's surprisingly limited research in this area. So in this project, we wanted to map out how the techniques used in pharmacological drug discovery could be used to develop a toxin discovery platform. And then we wanted to use our platform to find some potential target proteins and begin the process of target validation. Ideally, we want our toxin development platform to be reusable, so we could potentially develop different tools for different pests. So in the process of target-driven toxin discovery, as we're calling it, we need to filter and prioritize targets. So I've divided this pipeline into 3 components, although these are all interlinked. And these parts are, firstly, identifying groups of potential target proteins, secondly, comparing proteins between species, and thirdly, validating the targets. The first step is to identify groups of proteins that have potential as toxicant targets. And this is where genomes come in. So thanks to work largely by New Zealand researchers, we now have high quality genomes for almost all of our pest species, including stoat, shiprat, and possum. And we can compare these genomes with genomes from a variety of target and non-target species. The availability of these genomes is basically what enables us to do this project. So we're able to get clues about the differences in proteins with relative ease compared to traditional comparative physiological approaches. So essentially, this is a variation of genome mining. However, so we still want to narrow down the range of genes that we're looking at rather than looking at the whole proteome. And there are a range of different approaches to this. And I've looked at some different ways to do it. For example, I've been interested in looking at genes involved in key physiological processes like cardiac and vascular function. But we've chosen to initially look at G protein coupled receptors. And this is a family of proteins that have 7 transmembrane domains. And they're highly druggable, so many currently approved therapeutic drugs target GPCRs. GPCRs are involved in basically any physiological process you can think of, including cardiac and nerve function. They are well characterized, and there are a lot of small molecule ligands available that activate these receptors, so both endogenous and synthetic ligands. GPCRs are a good starting point, is that there's evidence of species-specific pharmacology, which can be mediated by subtle changes in amino acids, sometimes down to just 1 or 2. And this is often uncovered when people are trying to translate preclinical studies in model animals into clinical studies in humans. So there is potential here. So first, I need to extract out all the GPCRs from the variety of proteomes that I'm using, and there are a number of bioinformatics steps. But basically, I'm using the human sequences as bait to pull out lots of different potential sequences, and then grouping the receptors into orthologs. And these lists of receptors then need manual curation. Of the toxin discovery process is being able to compare proteins across species. And the simplest way to do this is across the whole amino acid sequence, using a pairwise sequence alignment versus the human ortholog. And although this is relatively straightforward, the data is still quite interesting. So it's reassuring to see that receptors from birds and marsupials are less similar to humans than mammalian receptors. And overall, we can use this to prioritize potential targets. But of course, what we really need to do is identify where the key amino acids involved in ligand binding and receptor activation differ between species. So another approach is to make a protein homology model of the predicted 3D protein structure based on the crystal structure of the human receptor as a template. So this is what I've done here, and I suspect this is the first model of a stoat receptor that's ever made, and it does give us the most information about whether amino acid changes or variations between species are likely to be near ligand binding sites. So the ligand here is shown in pink and orange, the amino acids. And while both of these strategies have pros and cons, this is basically a trade-off between how research intensive each process is compared to the accuracy that it provides. And there is an intermediate step where we can analyze just the conserved protein domains. And I'm working on this in case this is the sweet spot between these two other options. So we need to make sure that our new toxin has selectivity in the real world. So while we're looking at these very small protein differences, we need to keep in mind our end goal, and we need to define that clearly. So I've illustrated this with a hypothetical dose response curve, indicating how many animals are affected by a dose of a toxin. Ideally, we would have a clear gap between the response curves, so that a dose that produces a minimal effect on the non-target species would be effective against the target species. So this is similar to a therapeutic window. To work towards that real-world efficacy, we need to build up a profile of each potential target protein. This target validation involves collating a range of different pieces of information, some examples here. Fortunately, there are now a lot of really useful databases online, so we're using this information to build up cases for or against particular targets, and this gives us a really good head start. However, we also need to generate our own data, and we've made a start on the molecular pharmacology of our potential target proteins. So we've undertaken some preliminary work to express some possum GPCRs in a cell line, and we're then measuring the receptor activation in response to different compounds from a compound library to generate these dose response curves. And we can use this to see whether there are likely to be differences between possum receptors and receptors from other species. Is that the screening process also showed that activation of these receptors by some ligands induced cell death. And although this is a fairly indirect readout, it could give us some indication that the compound has potential as a toxin. So we're doing some more work here. Although this work is still in progress, what I hope I've shown you is that by using a strategic approach inspired by human drug discovery, there is great potential for new species-specific toxins. And what's exciting about this research is that we're drawing from a lot of different areas of research and building a platform that has broad applications for pest control. So thank you for listening, and thanks to Brian and Andrew, among many other people. And I'd like to thank Manaaki Whenua for funding this type of really super cool but quite early stage research. Thank you.